Hello and welcome to another installment of Ebatco Lab TV. In this video, we will look at scanning electron microscopy's functions and applications. Scanning electron microscopy, also known as SEM, uses an electron beam to achieve spatial resolution on the nanometer scale. Conductive samples are generally preferred for SEM, but with additional sample preparation, non-conducting and biological samples can also be observed in high-resolution images like these. So, how does scanning electron microscopy work? SEM actually isn't too different from optical microscopy. Instead of rays of light, SEM uses high-energy electrons to raster over a specimen surface. Visible light has a wavelength on a scale of 400 to 700 nanometers, while electrons have wavelengths around 1 nanometer to hundreds of picometers. This difference in wavelengths gives SEM a significantly higher magnification and resolution than our standard optical microscope. Similar to optical microscopes, SEM uses a series of lenses to focus the electron beam into a very small point, also known as a spot. When the electron beam reaches the surface of the sample, incident electrons interact with the surface to emit secondary electrons, backscatter electrons, characteristic X-rays, and more. A series of detectors are used to analyze these signals individually in order to produce a final image. The most common mode of SEM imaging uses a secondary electron detector, which detects low-energy electrons that are emitted from the sample surface. Secondary electrons only have the energy to escape from 5 to 50 nanometers within the material, making them very sensitive to topographical features. The result? A 3D-like image that is similar to observations made by eye. Sharp tips, edges, and taller features appear brighter in secondary electron images to give the realistic feel. Backscatter electron detection mode is used to detect high-energy electrons. Contrast in these images is primarily based on atomic number or elemental weight. Heavy elements appear bright, while light elements appear dark when analyzed in backscatter detection mode. Backscatter electrons have a penetration depth of around 500 nanometers, so some topographical information is still gathered from the sample. Due to its ability to look at both compositional and topographical features on samples, backscatter detection mode is great for looking at compositional variability and at the microstructure of materials that are composed of different elements. Backscatter electrons aren't the only way to gain compositional information about samples. During secondary electron emission, which is an inelastic scattering event, X-rays are produced which have an energy specific to the elements within a sample. These characteristic X-rays can be used to determine the element types and quantities present in the sample. This technique is called energy dispersive X-ray spectroscopy, or EDS. EDS can produce a spectrum of all elements on a sample at a point, or can be used in tandem with SEM to produce EDS maps that display the presence of each element on the sample surface. SEM is a multifaceted tool that can be used to understand multiple different technical parameters. Since the stage can rotate and tilt, it's possible to determine the surface roughness by superimposing two images, each with a different tilt. This gives information on the average roughness, root mean square roughness, and the maximum peak to valley roughness. For failure analysis, SEM images are ideal to identify fracture modes, as well as crack initiation and crack propagation locations by analyzing a material's microstructure. SEM data can also be analyzed using image processing software to perform grain size, particle size, and pore size analysis at the micro and nano scale. Scanning electron microscopy is one of the most powerful techniques to analyze samples for a variety of material properties. It is used in biological, geological, medical, manufacturing, semiconductor, and many more fields. Secondary electrons give topographical and morphological information on samples, and backscatter electrons capture compositional information as well. SEM can also be paired with EDS to perform quantitative element identification and chemical analysis. That's all for this installment of Ebatco Lab TV. We hope you'll visit us again. If you enjoyed this NetLab video, please leave a like and subscribe. Keep an eye out for more videos covering diverse topics from our lab. If you have any questions about either the methods or instrument you saw here today, don't hesitate to give us a call or shoot us an email. And as always, if you're interested in microscopy or other materials analysis lab services or consultation, please give us a call. We'd love to hear from you.